Okay, say it. Can you repeat the question, please?
So my question is, why does a lined up sequence for an alpha helix give you one hydrophobic face? This was also covered in chapter 4 too. I think a couple slides before the Ramachandran plot, maybe.
Before?
Yes, we're talking about alpha helices.
Right. So obviously you want in the alpha helix, you want the so what keeps it all together is these hydrogen bonds between the NH and of the amide and the CO of the carbonyl, right? They're all shown here in dashes like that. And the aliphatic groups are on the outside. And obviously you want as much interaction here as possible, right? So you want these two groups to be interacting. So that's why we say like n+3, n+4, right? So this is 3.6 residues. So they're not aligned directly. They kind of, I think they slightly misaligned, and that's why it's kind of found halfway towards the turn. So it's 3.6 residues, but they're still close enough for them to be interacting either favorably or disfavorably, depending on if they're for example, if they're charged, right? You want plus and minus for the attraction, so that'll stabilize. If it's two minuses like aspartate, for example, or glutamate, right? So those are going to be minuses. So you don't want the aspartate and glutamate in these positions. And you don't want two lysines in these positions, but you want lysine and aspartate, for example. So what was your question about the alpha helix?
Why does the linear sequence give one hydrophobic face? Because on the next slide, that was a question you asked.
What kind of sequence? Yeah, so I gave it to you, right? So this must have come from the lecture. So valine, leucine, isoleucine. So if we go back, right? So this will be valine. This will be leucine. This will be isoleucine. Right, and so this entire face on the right-hand side of the helix, this will be hydrophobic.
Okay, that makes sense. Thank you.
Yeah.
And to finish up, my last two really quick questions. Will we be looking for any free energy funnels on the final?
Free energy.
Yeah, it's pretty much just talking about how like the intermediate motifs have to overcome to get to the native fold.
Yeah, so basically anything we've discussed in class will be on the exam. If we did not discuss in class, I would give you pages to read. So if it's neither of those, then don't worry about it.
Okay, so I think that answers number 4, that expected research paper we've covered in class too.
Yes.
Okay, thank you so much.
Mm-hmm. You guys have questions? Devin, you always have a question. You're muted.
Yeah, I did. Can we go back to the Ramachandran plot?
Uh-huh.
So you said if there was like a question, we would have to say, oh, like, oh, what's the range that is favorable? So like, would you say, oh, what's the range for a right-handed alpha helix? And we would give 60, 57, if like that was an option.
No, it will be drawn for you. So yeah, I will not want you to memorize 47 and 50.
Oh no! I meant like we would have this, and
Yes.
we would say -60, -57.
Yeah.
Okay, but we need to know right-handed helix is in this region, and anti-parallel, parallel are in this region, right?
Yeah, I don't think there's so much. I mean, you just roughly you need to know where right-handed helix is and where the beta sheets are. Yeah, they're
Okay.
up there.
I remember hearing that you said on Friday that we should review the sapling and the previous
Yeah.
exams, but we can't review the previous exams.
Previous exams, yeah, you can't really review previous exams, but
Like, are you going to pull questions from there?
no, I'm not gonna do that. So,
No.
so I'm not gonna take questions from the previous exams and put them on the final, which I already made the final, so I have not done that. So no.
Okay.
Is there any way that you can open the previous exams for us to just kind of look at them?
Yeah, if you want to go over your exam again with me, that's fine. Just we can do this separately. Yeah.
Are you going to be pulling any exam questions from sapling and like converting them? I know there was a few from the first exam that had that. Like
Yeah, what I usually do, I go through SAP and see if there's anything that looks like we discussed in class and it's pertinent. And I usually change them. Yeah, I don't take them straight from sapling. I just change them in one way or
Yeah,
another.
that's what I was asking.
Mm-hmm.
Okay. Cool.
Sapling has great questions, and the only problem is if I don't change them, those sapling questions you can Google them.
Yes.
So that's why I changed them.
I think. I'm trying to remember my other question. It had to do. It's like from exam 1 material. Like having to calculate like KDs or KAs. Do you know which slides those are? I think it's from here. I have my copy.
It's chapter 4.
Chapter
4 or 2. 4 or 2. 4?
4. It should be. It's
So is this chapter then?
Is it? Oh, sorry, 5. Sorry, 5.
Yeah, it's 5.
Yeah, that's
Oh yes, 5. That's right, 5. You want me to open that one?
Yeah. Yes, please.
And while you're pulling that up, a quick question about the free energy funnels. Like, how would a question look for that be structured? If that makes sense. Like, we're just supposed to kind of like interpret what the intermediate is supposed to be like for the motif form.
Okay, sleep peacefully. I'm not gonna ask you that.
Okay, thank you.
All right, so 5. So association constant, dissociation constant, Ka and Kd. So
Okay. I don't know.
So fraction of occupied binding sites. Yeah. So this is, you need to know this one. Yeah.
Which one, the red or the? Or the blue?
Well, this equation. Yeah, you need to know this equation.
Okay.
But in general, have an idea what the association constant is and what units are used to measure it. And
Yeah, because I remember that unit question from the first exam, and I didn't
Yeah.
study my units, and that ended up getting me.
mm-hmm.
You have that unit list on this PowerPoint, right? I think so. I think it's at the end. We don't have to pull it up. I just wanted to know so I can study it this time.
And so we talked about strong binding and weak binding.
Yes.
So
And we
now
cover that again when we talk about like Michaelis constant, right, and all that.
Yeah. Mm-hmm.
So a really small KD, it means tight affinity, and a really big KD means less affinity.
Mm-hmm.
Obviously, we need to know all the structures for, like, glucose, galactose, maltose, and how they interconvert in between. Is the list that you gave us sufficient still? That list of
Yes. Mm-hmm.
Okay, cool. So I want to make sure. I think that's all I have for right now. Anyone else wants to ask anything?
Sorry, could you repeat the thing about affinities?
David, you want to do it or you want me to do it?
Yeah, I can do it. Yeah, so basically, if you look over here at the, so when strong binding, your KD is going to be really small. So the way he did it on exam 1 was he asked, like, there was, okay, 1.0 times 10 to the negative something. And those units, so it's like nano, pico, milli, femta, stuff like that. The smaller you get, so if you have 1 times 10 to the, to the second, compared to 1 times 10, 1 times 10 to the negative 2, compared to 1 times 10 to the negative 5, the negative 5, that's going to be a smaller number. So your KD is going to be a lot smaller. So and KD is your dissociation constant. If you have a really small dissociation constant, that means that your binding affinity for that substrate with that enzyme is going to be really small. No, it's going to be really big. So it's kind of like an inverse relationship. If your KD is small, your binding affinity is big. It's going to be strong.
Yeah. So the smaller this number is, the closer to unity this approaches. Theta.
Perfect. Thank you both so much.
So theta, if it's 1, so theta is between 0 and 1, right? So if KD is very small, then your theta is 1, which means that your binding sites are fully saturated. So it has the highest binding, tightest binding.
Thank you both very much.
Mm-hmm. More questions?
Yeah, since we're on the slide, could we go over the mechanism here with with histidine 93 and histidine 64, and like the difference between what's going on here? Or do we not need to know that for the exam?
I think I remember. I don't remember myself. I guess I did. This is something that I something that you need to review constantly to remember.
Yeah, because one of them,
So
they both do something specific.
yeah, so histidine, 64 is the histidine that binds iron, right? So the iron is in the middle of the heme. Well, let me see. Hold on a second. Maybe it's, yeah, hold on a second. Myoglobin. Yeah, it doesn't really explain it here.
Isn't it his? Isn't it
What
histidine?
if I will not ask you that level of detail, obviously.
Oh, really? Okay.
Yeah, I think you're overdoing here. What I may ask you, for example, is what amino acid is iron bound to in the myoglobin?
Okay. That's his.
And then you will have to say histidine. And so it's I'll if this is my level of knowledge, then that I don't expect you to to know that detail, level of detail.
Okay. So, and I've seen this with a few other finals with the higher level like biochemistry classes. The finals are usually like, getting more general idea stuff and not getting so much into the nitty gritty, kind of like what we're talking about here. Because I remember in the sapling having to differentiate the function of 93 between 64.
Yeah, I do remember that.
But so the final, we're doing more general, not so much nitty. There will be probably some nitty gritty stuff, but more general, like what you're talking about here, like, oh, it's just histidine.
Right. But by doing that problem, you will definitely remember that iron is bound to histidine.
Yes. Yeah, definitely.
You may not remember which one, but you will remember that it's histidine.
Okay.
And it will also help you remember what histidine is, right? So by looking at the structure of histidine, imidazolurine, it will help you remember what amino acid it is. So all these problems are tied together, they're connected.
I have a quick question while we're on the slide. Could you go over how the dissociation constant turns to a partial pressure in terms of binding oxygen?
Well, it's for gases it's partial pressure, for liquids it's concentration. So so what is KD basically? KD, if you think about the definition of KD, would be a theta, the saturation, right? Half of maximal, so 1/2, right? So if 1 is maximal, so basically KD, one way to define that would be it's the ligand concentration at which theta equals 1/2. This makes sense? So just plug in, just take KD out, put the ligand in, and what do you get? You get 1/2, right? Can you see that?
Yes.
Right. So and so the P50 is the same thing. So basically it's the pressure of oxygen at which half of the binding sites on heme have been occupied.
Okay, so it's just, okay, I see it now. Okay, thank you.
Yeah, it's right here. When ligand concentration equals KD, then theta is 1/2. And you can get the KD from the graph, right? So 0.5, extrapolate, and you can get the KD.
Gotcha. Thank you.
Mhm.
Or you can say that one more time. E equals 1/2.
Yeah. So KD is the substrate the ligand concentration at which half of the binding sites have been occupied. And so you can calculate, you can get it from the graph, right?
Half of. Okay. So this is very similar to just like, I forget which chapter it is, but where KD equals the substrate constant, like half the substrate, like with Vmax and everything.
Yes. Mm-hmm.
Okay.
So the Vmax, so there Km is the substrate concentration at which the enzymatic rate is half maximal.
Okay. Mm-hmm.
And here is the half of the binding sites have been occupied. So how is the studying going?
Pretty good. Like at this point, like everything makes more sense as like reviewing it because other classes definitely helped me out in understanding biochemistry. So as I'm reading, like, oh, I know this, and I questioned why I did so bad on the first exam, but you know, it's over.
Mm-hmm. Cool. Yeah, that's why you have these prerequisites, right? So that so that you can utilize the knowledge from your other classes and make connections.
I've got a quick question. I can't remember if this is like chapter 2 or 3, where we had a bunch of different like chromatography that we were looking at, like size exclusion chromatography, affinity chromatography, and then ion exchange chromatography is the one that I always forget exactly if the resin is positive or negative on a cation exchange and what it is on an anion exchange and which one elutes first. Like I get tripped up on the cation exchange. If you don't mind going over that or the ion exchange ones, if you don't mind going over that again, that'd be great.
Okay, so now we need to remember which chapter that was, right?
It's chapter 3.
Yeah, chapter. Yeah, I was about to say.
Awesome, thanks.
Okay. All right, so all right, so obviously we're going to separate the you realize, right, so how many proteins are present in a single cell. So to isolate a protein of interest, you basically like it's like a fishing expedition, right? You fishing for a tiny little thing in the ocean of proteins.
Right.
So generally, you apply many different methods to purify proteins using their physical and chemical properties. So charge, size, affinity for ligand, solubility, hydrophobicity, and so on and so forth. So probably it's not given here, but one of the first thing you normally do is you apply a technique known as salting out, which is just based on solubility, right? So many proteins are water soluble, many are not. And so, and if you, when you add salt, it becomes much more ionic. So if you think about water is already quite polar, right? So if you add polar salt, the water becomes even more polar. And so you can separate the proteins based on their polarities that way. It's not very specific. So if the two proteins have the same polarities, they will stay in the solution or precipitate at the same time, right? So salting out is the first thing you would do. And then once you've done that, so you collected the precipitate, or you decide to work with the aqueous fraction, then you do column chromatography. Column chromatography, so this is basically, there are different kinds of stationary phase that will interact with proteins separately. Or with each protein, the interaction will be individual. And you can see here, so this is column chromatography. So we're gonna have the stationary phase, so we're gonna have a liquid phase. And remember, this is based on distribution of the protein between stationary phase and liquid phase, right? So you wanna do it slow enough so that you allow for the protein to equilibrate between the aqueous phase and stationary phase. And so in this particular case, it was made very easy for us because the proteins are colored. So we can actually separate them based on color, but or visualize them based on color. But in most cases, they will not be colored, in which case we need to have a detector. Detector can be a UV detector, for example, or can be MS, a mass spectrometer, can be a GCMS, LCMS, and so on and so forth. So this is column chromatography. So it's just a general way, depending on how you design your stationary phase, you can separate different kinds of mixtures of proteins. Okay. Then you ask about ion exchange. So let me actually close this. So ion exchange chromatography. So here, the resin will have a particular charge, okay? And usually when it's negatively charged resin, you have some kind of sulfate residue. So let's say this is the polymer. And here you will have negatively charged residues, right? And so you can see these beads, right? The negatively charged beads. And so the protein will, obviously some parts of the protein will be positively charged, some parts of the protein will be negatively charged. But in general, if you have more positively charged sites on the protein, it'll stick to the bead much more strongly, right? And so so basically, again, you run your proteins down the column, right? And you can see here, if the beads are negatively charged, then the proteins that have a lot of positive charge, the blue ones, right? They will barely move. They will barely move. Whereas the red ones, they will have a primarily negative charge, which means they will be repelled by the resin, right? So negative hates another negative. And so the resin will repel them. And so these will not go into the stationary phase at all. These will be pushed through the column with the eluent, with the solvent. And so the first fraction that you're going to collect will contain primarily negatively charged proteins. then neutral, and then positively charged.
Got it. So cation exchange has a negative resin, and so the negative charges come out first. Okay.
Yeah, right. So this is something that I always like to trick students. So it is a cation exchange, even though that the bead has a negative charge.
Got it.
Right. So it's a little counterintuitive.
It is. Yeah. Okay, great. Thank you.
But there are others. We can keep talking if you want.
Absolutely. Sure thing.
Size. Then size is important, right? So so here the pore, so basically the stationary phase contains a porous material, right? And only small molecules can actually get inside, whereas large molecules will not be able to get inside these pores. And so what will happen, the proteins, the large molecules will elute first, right? And the small molecules will stay in the stationary phase because they got stuck in these pores. And so they will, again, it's an equilibrium process. They go inside the pores, they get out of the pores, and back and forth. But it results in a slowing down of the movement down the column. So I think these are, I would say, and the binding affinity. So this is an important one. If you have a ligand, right? If you have a ligand, you can actually attach your ligand to a bead. So this is the small spheres are ligands for particular protein. And so the protein can bind to these ligands, and by doing so, it will get stuck in a column as well. It will get stuck in a column, and so basically you have a column, you put the ligand in, and then you run your mixture of proteins through, and only proteins which are specific for that ligand, only those will get stuck in a column. The other ones will just wash straight through. Does this make sense?
Yes, thank you.
Yeah. And then the final step is actually critical. So how do you get it out? How do you get it out? You have to apply the ligand, soluble ligand by itself. Okay. So in other words, something that will compete with the beads. Something that will compete with the beads, and it will take the protein down.
How did that make sense?
Yeah.
I have a quick question. Going back to chapter 5, if we could go over the models of cooperativity, I can't say the word, oh my gosh, cooperativity of the concerted and sequential models.
We did not cover sequential.
We didn't?
No.
Okay.
And I did not include it into the final. You said chapter 5?
Yes.
Hold on a second. Maybe I'm getting confused. Hold on a second. So what are you talking about?
Cooperativity. Right there, 26.
Oh, yeah. We did talk about that. Yes. Cooperativity. Mm-hmm.
So can we go over like what the model means? Because I was a little confused.
Well, I mean, again, just like a question by Devin, I do not want you to know all these little details as far as various possibilities here. I just want you to know that what this allosteric protein is, right? And how does the allosteric proteins work? So in other words, I want you to know not this slide, rather I want you to know this slide.
Gotcha. Thank you.
So you can see here, so there are two binding sites, right? And so when the ligand binds to one of these sites, that affects the other binding site and makes it more hospitable to the to accept the ligand, right? So the first event facilitates the second event. That's why it's called cooperativity. Is so you can consider this as an allosteric protein.
Okay.
You know what allosteric protein is, and you know the major how it works, right? And in other words, if you can describe to me this figure, then that's all I need.
Okay, great. Thank you.
Can you zoom in on this figure? Okay, so you said that the binding sites, those are allosteric. And so that's why they're, what am I trying to say?
Mm-hmm.
When you have the ligand bind, it causes shape change. It causes a shape change in all the rest of the parts because there's the four parts of the heme.
Yes. Mm-hmm. Well, here it's only two sides, but
Yes.
in the hemoglobin it's four. Yeah.
And this was slide 25.
Yes.
Cool. Thank you. Oh, I have one question actually. I forget which chapter it was, but I have some flashcards right here. So it was like, okay, endoprotease, protease, lysine C, what does it cleave? And like, what does trypsin cleave? What does submaxillary protease cleave? Like, and they, some cleave like lysine, arginine, bonds like that, like chymotrypsin. We need to know, obviously we need to know that, and lysine. But do we need to know all these other ones like endoprotease, cyanogen, isoelectric?
No.
Oh, sorry, that's a different one.
No.
No. Okay. But we do need to know what chymotrypsin cleaves and.
Yeah, that would be good. Mm-hmm.
Yeah, like and those are aromatics.
Yeah.
Okay. And lysosome, or is it lysine? No, lysosome's a organelle or.
Organelle. Mm-hmm.
But lysine, we don't need to know what that cleaves. Like, what are those in tiers that we talked about?
Trypsin, right? You said.
Wasn't there another one? Started with an L. It was chymotrypsin, and then there was another one that started with an L.
Is there a slide for that?
I
don't know.
Yeah, it's a little bit after the chymotrypsin one, I believe.
I don't know if we actually paid too much attention to these.
Okay.
Which do you know chapter?
I think it's chapter 3.
Is it chapter 3?
Yeah.
Oh yeah, for the yeah, I got it. Yeah, the reason because we didn't go over that in class. But I assigned reading, right? Didn't I assign reading?
Yes.
That's where you got it. Mm-hmm.
But focus more on the chymotrypsin than the other one. Is what you're saying.
But I mean, there are couple specific proteins. That's yeah. You can just remember those.
Okay.
I had a question about, I think it was chapter 9. On the exam, the past exam, you asked about a data set that would be obtained from restriction
Yes.
digests.
Mm-hmm.
Can you go over that a little bit?
The exam question.
Yes.
So so these are various restriction and the nucleases, right? So there are two of them. A fragment of DNA that's going to be cleaved, and so I think it's pretty much straightforward, right? So if this works on this sequence and cleaves it at this position, then you get two fragments, 2 in 8 kilobases, right? So this is in thousands, so it's a kilobase pairs, right?
Okay.
So this one will be 2 in 8. So eco R1, then BAM H1 is gonna cleave it in half, 5 and 5, 5 and 5. And then if you employ both of them, first you get the two 5s will be cleaved. 2 5s will be cleaved, and another side will cleave it into, one of the 5s will be cleaved into 2 and 3, 2000, 3000.
Okay, got it. Okay, it makes, I don't know why on the exam, it like really, the last column, the eco RI and BAM HI like tripped me up a little bit, but okay, I understand now.
Mm-hmm.
All right, thank you.
Continue with the exam before you close it. Can we go to the one that asked about, it was a long chain of amino acids and talk about something about being hydrophobic?
A long chain of amino acids.
It was like it was just like which one is like more likely to be something about being hydrophobic or something like that. It was on the exam.
Well, this is a different exam actually.
Oh, okay.
Yeah, that's not your exam. Oh, боже. Yeah, I don't think this is the right exam. This was the last exam?
Yes.
Oh, I remember the one you're talking about.
Yeah.
Hmm. Yeah, that one I remember looking at it being like, what? And then I was like, oh wait, it's amino acids, and then having to figure them all out one at a time.
Yeah.
Question is, how do I?
Preview.
But preview is gonna give me one question at a time.
Oh,
Let me see. Let me see.
well, you could go to edit and then turn off one question at a time, and then
From edit, I can do all questions. You're right. So
For me, it was near the end. I don't know where it is in this sequence.
it's at the very end. Oh, maybe it randomizes them.
Yeah, it's shuffled.
so we missed it.
I think so, because there's that one. What was the question? It was something about which one is like most likely to be hydrophobic or something like that. If I saw the answer choices, I would know, because like that was like, it was just long lines of amino acids.
This.
Yes.
Mm-hmm. Yeah. So which of the following segments of the integral membrane protein glycophorin most likely contains membrane span and sequence? Yeah. So you're looking for hydrophobic sequence.
Mm-hmm.
And so here are all the non-polar, all hydrophobic amino acids. Valine, methionine, alanine, right? Leucine, isoleucine, phenylalanine. Now this one, so that's the correct answer. This one has tyrosine, which has a hydroxy group. K is a lysine. S is serine, has a whole bunch of hydroxy groups, right? So that's not gonna work. Now here's acids with negative charge. Threonine has the hydroxy groups.
So the reason why I asked about this question, go ahead.
Carboxylic acids all over the place. Yeah, and these are lysines all over the place. Yeah, so just basically look at what kind of amino acids and you select the one that has most hydrophobic content. Yeah.
Okay, so we're looking for majority. Okay, that's because I was like, because it took me a while on this question because I was because I was kind of like, there's some threonine in like the correct answer. So I'm just like, so that's not completely hydrophobic. So, okay, it's majority. That makes sense.
Yeah, I see what you're saying. There's threonine here. Mm-hmm.
For that one, would the order of the amino acids also matter, just for hydrophobic sake?
You mean like maybe between T and T, right? Maybe it crosses the membrane between T and T. In other words, looking for a longest stretch. Yeah. And here the threonines are kind of cutting the whole thing in half, basically. But they also have lots of charged ones, right? Lots of charged ones. I think once you see the charged ones, then you can immediately disregard that. You know, you can't have a charged amino acid in a membrane.
Basically, what I did for this one is I went through each one and kind of just marked, okay, I marked all the non-polar species and just basically just chose the one that had the most non-polar. Because this one, like when you mark them all out, this one had significantly, like almost double the rest.
Yeah, makes sense.
Okay, good strategy. Thank you. It was just like time crunch, and then it's like, whoa, wait, I got what 20 amino acids to go through. So definitely a time crunch thing.
I remember you saying, I think it was last class, that the extra credit that we did, that was very, you said that was going to be similar to like the types of questions we're going to be getting on the final. Is that correct?
Sorry, can you repeat this again?
Yeah. I remember you saying on Wednesday of last week, Wednesday this week? I'm getting all my days mixed up. That you said that, so the extra credit that we did, that is, those types of questions are the ones that are going to be similar to what's going to be on the final, more like general idea.
Yes, yes. Mm-hmm.
Okay. Just wanted to make sure.
All right, guys, we'll keep studying. Or for most of you, just a review at this point. You already studied for this once. So, and throw some ideas into discussion section, discussion forum. So, yeah, I'll certainly be monitoring those.
Okay, thank you for having this review session. Like, this is amazing.
Yes, thank you.
Sounds good.
Thanks so much.
Thank you.
Mhm.
Dr. Kornienko, I actually had one last thing. So I know that sometimes during the exams, I have like questions on like clarification. Is there any way we could have like Zoom open, like you there, so we could just come and pop in and ask a clarifying question if we need during the final? Because I know like when we have just one question at a time, having to wait for an email response, because I know you're probably getting a lot. I can't move forward usually on a question until I get the clarification.
Yeah, we can do that. Mm-hmm.
Awesome. Thank you very much.
All right, guys. Have a productive weekend.
Thank you, you too.
Bye.
Thank you.
you.